So the last macromolecule are the nucleic acids, and these ones are kind of hard to group into a category like carbohydrates or starches and breads and proteins or beans and meats. Nucleic acids are the hereditary material. So this is the DNA and the RNA inside of the cells. So this means that everything you eat that has DNA or RNA has nucleic acids in it. This is not like a nutrient generally, but it's still a important macromolecule. So nucleic acids are organized into hereditary units called genes, and these genes actually code for the proteins that we make in our body. And the main two types are deoxyribonucleic acid. This is your DNA. So this is what's found in the nucleus of our cells. This is what is our genetic template. This is our genetic instructions for the entire body. And then we have something called ribonucleic acid, which is similar to DNA but has some structural differences. And this is not necessarily found in the nucleus. There are different forms of RNA. So we have messenger RNA, which is something that sounds like a messenger, takes information from the nucleus, brings it out into the cell. And we also have other types of RNA, transfer RNA, ribosomal RNA. We'll talk lots of different types of RNA throughout the rest of this class. And the flow of information through your body can be summarized as what we call the central dogma of biology. And this is that information starts as DNA, it gets transcribed into RNA, and then it gets translated into proteins. So we start with DNA, then RNA, and proteins. And you should memorize this now because it's going to come up later and it's going to be important. So DNA, RNA, protein. Say it with me. DNA, RNA, protein. So here's a bit of an image of what's happening. This is the nucleus of the cell. So the DNA lives inside the nucleus. The DNA is huge, and it's this massive like double helix coiled up upon itself. You have 46 chromosomes, so there are 46 pieces of DNA in your nucleus of every single cell. And when we want to make a protein, what we do is we copy a small portion. We copy one gene into a piece of messenger RNA. That messenger RNA is smaller, it's just a single strand. It's able to leave the nucleus, come out here to the cytoplasm, find some ribosomes, and then start to form a polypeptide. So each of these little purple circles is an amino acid, and this is how we build proteins. So we'll start with just the basics of nucleic acid for this chapter, and then when we get to genetics in the end part of the semester, we'll go into these in a lot more detail. The monomer is the nucleotide. So this is my monomer. And each nucleotide has three important parts. It has a nitrogenous base, and it is nitrogenous because it has nitrogen in it. It has those amino groups that makes it a base. It has a 5-carbon sugar. So here is my sugar. We call them a pentose sugar. Penta means 5. So there's 1, 2, 3, 4, 5 carbons on that sugar. And then they have a phosphate group. So there's that functional group, the phosphate group. So you have to have 1, 2, 3 of these things to create a single monomer, which is a nucleotide. So then we put multiple of these nucleotides together, and it makes the polymer, which is the polynucleotide. So this just means many nucleotides. There are 5 different nitrogenous bases. 4 of them can be found in DNA, and 4 of them can be found in RNA. A pyrimidine are the 3 that only have a single ring. So I like to remember this as the long word gets the small structure. So they're opposites. And then the purines are the ones that have 2 different rings. So this is the shorter word, and then it's got the larger structure. That's how I remember purines and pyrimidines. So DNA is going to contain adenine, guanine, cytosine, and thymine. So adenine, guanine, cytosine, and thymine. And then RNA has adenine, guanine, and cytosine, but instead of thymine, it actually has uracil. So for this right now, I want you to know the 3 big differences between DNA and RNA. So the first one is the sugar. DNA is named deoxyribonucleic acid because its sugar is called deoxyribose. So you can see that pictured right here. RNA is ribonucleic acid because the sugar that makes it up is called ribose. Those are both 5-carbon sugars. So ribose, you can see right here, has an hydroxyl group. And then the only difference between ribose and deoxyribose is that it's missing that oxygen right there. So it is literally one less oxygen than ribose. That's the difference. The structural difference is really, really small, just one difference of an oxygen. Nitrogenous bases are different. DNA has thymine, whereas RNA has uracil. So you'll never see a thymine in an RNA, you'll never see a uracil in a DNA. DNA is also what's considered a double helix, so it's kind of 2 strands woven together to create what's called a double-stranded helix. RNA is typically 1 single strand. Sometimes these strands kind of fold back on themselves, but either way, it's usually only a single strand, and we have lots of different types of RNA, and so you'll see these strands in lots of different configurations depending on what kind of RNA we're looking at. So the way we build polymers, again, is by linking nucleotides or the monomers together to make larger molecules. And so you can see here we've got the phosphate group, the sugar, phosphate group, sugar. So right here, we've got a covalent bond between 2 monomers. And in the nucleotides, in the nucleic acids, this is going to be called a phosphodiester linkage. This is going to form between the sugar and the phosphate. And so what happens is in a strand, so this is just 1/2 of a DNA molecule, the nitrogenous bases, the A, T, G, and C are all on the inside, and then we have the sugar phosphate backbone here. And so that creates what's called a backbone. And then these nitrogenous bases, they're calling them kind of like an appendage. If I had another strand over here, here's my backbone, sugar, phosphate, and then the nitrogenous bases would be inside. DNA is going to abide by some complementary base pairing rules. The width of a DNA molecule, so assuming that I untwisted the helix, this width has to stay consistent. So a single ring structure has to always bind with a double ring structure, and that's going to keep the width consistent. So specifically, adenine is always going to bond with thymine, and guanine is always going to bond with cytosine. And these two are going to be held together with hydrogen bonds, and I just drew the number of lines to represent the hydrogen bonds holding those together. So this specific pairing rule is what makes copying DNA so accessible. I can have a single strand of DNA and know the side A, T, G, C, C, T, A. Right? So if I have one single strand, I can make the other strand without any other information. I know that A pairs with T, T pairs with A, G pairs with C, C pairs with G, C pairs with G, T pairs with A, and A pairs with T. So I can come up with the complementary strand from just a template strand. So this actually makes it really easy to kind of split DNA into 2 sides and replicate it so that our cells can divide and have more DNA. So here's the overview of the nucleotides. You need to know the basic structure of the nucleotides, the 3 parts: nitrogenous base, sugar, phosphate. You need to know the 2 different examples, DNA and RNA, what their functions are. That covalent bond is the phosphodiester linkage.